Hi everyone, my name is Nanthia Suthana from UCLA. I'm an assistant professor in the David Geffen School of Medicine at UCLA and the Semel Institute for Neuroscience and Human Behavior. Today I'll be talking about deep brain recording and stimulation of real-world episodic memory in humans. So of course, memory disorders are a huge problem today, and in several cases such as Alzheimer's disease, that most people are aware of, where we have over 5.8 million affected in the United States alone, with $1 trillion annual worldwide costs and increasing. Of course, there are also memory impairments in patients who have had traumatic brain injuries, epilepsy, and various other neurological and even neuropsychiatric conditions, for example, in post-traumatic stress disorder, where here the memories, traumatic memories are, wanting to be suppressed in this case, whereas in Alzheimer's disease, it's an issue of amnesia and forgotten memories that they're episodic in nature and personal. So of course, these rates of Alzheimer's disease, as I mentioned, are increasing over the next 30 years, where the costs are also increasing in addition to the number of people affected. The areas of the brain that we know are implicated in disorders of memory are in the deep brain areas, in an area called the medial temporal lobe. And we know this from decades of research working with many studies, but in addition to patients, rare patient studies such as this one here, patient HM who had epilepsy and surgically had his bilateral, so two sides of his medial temporal lobe removed. You can see an MRI of his on the left-hand side compared to an unaffected individual where those structures are still intact. Now, HM was unable to form memories for new everyday events. And this type of memory, known as episodic memory, is one of the first to be affected in patients, for example, with Alzheimer's disease. The regions that are important within this medial temporal lobe, you can see on the right if you look at a side view of the brain. And these regions include an area called the hippocampus and entorhinal cortex. Now, HM had these areas removed as well as some additional adjacent areas, but we now know that these areas are very important for being able to form memories for events in your life. About 8 years ago, we published a study showing that if we used electrical stimulation delivered to this entorhinal area, which is thought to be the major input into this entire circuit for memory, was sufficient to enhance memory in a small cohort of patients who had electrodes already implanted into those areas for evaluation of their epilepsy. Now, we've since followed up on this study and found that in these patients, when you deliver electrical stimulation, that's very, very low to where the participant is unaware that it is being delivered. You can see an enhancement of memory in those areas, but the location of that electrode stimulation matters. And it turns out that a few millimeters off can make all the difference, where you see here an increase in the purple of memory performance when the stimulation is in an area called the angular bundle, which is thought to be the white matter area of this entorhinal area. As opposed to on the left, you see gray matter stimulation was not sufficient to show any kind of change in their memory scores. This was all within the same participants. So in some trials, the images or the stimuli the participant was trying to learn, stimulation was delivered either to a gray matter area in the entorhinal area or into the white matter area. And it turns out this white matter area, which consists of the connections between this area and the hippocampus, was the important key region. And this just shows you single participants, each line showing a participant with on the right side in purple showing the right-sided stimulation, and a positive slope indicates that memory was improved. And you could see all but one participant showed an improvement in memory with stimulation. Now, one thing to note here was that the electrodes were placed in the entorhinal white matter area, and we wanted to see whether this was true for other types of memory tasks, not just learning of individual faces. So we did a series of tasks where participants had to learn images of objects, like here bicycles or people or face name pairs. And we saw that the location of the stimulation mattered in all of these cases, such that if the stimulation was to the right side in the white matter of the entorhinal area, you saw an improvement in memory, so shown in purple, whereas you don't see this effect in other areas, such as the gray matter. Here shows just an example of where these electrodes are placed. If you look at an MRI scan straight ahead, coronal view, where the little red dots are the location of stimulation, and shown here is an example on the top where the location is in the gray matter of the entorhinal area or cortex, and the bottom is in the white matter. Again, if you look across all of this data, across dozens of participants, you see the location matters in that the right-sided white matter seems to play a role in enhancing these types of memory. Now, all of these tasks, the participants were learning images on a screen, on a computer screen, whether they be faces or objects, or in another task that I'm not showing you here, words. Whether this type of memory really translates to the real world when you're learning about events in your life, what you had at a dinner party last week, who was there, all of these types of information that you're learning on a daily basis, whether this type of finding can translate to the real world is unclear, because we don't test real world memory in the laboratory. We usually test memory for these two-dimensional images on a computer screen. Now, a lot of the work that's been done in the field of memory has been done in rodent studies, where actually the Nobel Prize was awarded to 3 individuals in 2014 for the discovery of brain cells that may be implicated in memory, episodic memory. And the way that they study memory in rats, and mice, is through spatial navigation. So we have animals run around in a maze while these cells are being recorded. And the finding that won the Nobel Prize was that there are place cells in the hippocampus and grid cells in the entorhinal cortex that will fire when animals in particular locations across the environment. Place cells, as the name suggests, respond when an animal is in one particular place, whereas grid cells fire in a grid-like pattern, where you see here a couple examples where there's this nice repeated pattern across the circular environment that looks like a grid. And it's a hexagonal grid that has a nice 60-degree geometric pattern to it. Now, these cells are found in this area that we know is implicated in episodic memory, that's early affected in Alzheimer's disease. And the other finding that's important here is that there are theta oscillations, or brain activity waves, that occur about 8 times per second. You can see one from a rat down on the bottom. And these are very tightly coordinated to these place cells and grid cells, and are thought to be important for learning and remembering these episodic type memories, that in the case of animal studies is necessary for spatial navigation. As I told you before, the way we study episodic memory in humans in a lab is to show them two-dimensional stimuli. We have not been able to replicate these findings in rats because humans are not running around in mazes while we record their brain activity. And so instead of these theta oscillations, we found these lower frequency waves, which you see here on the top right, which is about 3 to 4 cycles per second. And so we've wondered during these years, whether these findings in rats could translate to humans, and whether our findings in the laboratory of improving memory with stimulation could also translate to the real world. So of course, this is really challenging in the human brain because, as I said, recording from the human brain requires that the participant is stationary and is not moving. So traditional methods that are shown here, such as MRI, functional MRI, MEG PET, and even recording from electrodes in patients with epilepsy, requires them to sit still because of motion artifacts. And the other limitation here is that these techniques can only record and or stimulate superficial brain areas close to the surface of the head, and can't reach these deep brain areas, such as the hippocampus or entorhinal cortex, that we know are important for episodic memory. The third issue that I already mentioned before is that the tasks that we use in the laboratory involve two-dimensional stimuli, which are not naturalistic or are ecologically valid, and so whether our findings translate to the real world, that is unclear. However, we've had a recent opportunity to take these type of studies out into the real world, and that's with the development of a new neural implanted device called the responsive neurostimulator. You could see here on the top left an example patient with a electrode that is implanted in the hippocampus, deep in the brain. And these electrodes are implanted for treatment of epilepsy. And so patients have these implanted and they record seizures and stimulate on demand when the device detects seizure activity to hopefully prevent the seizure from occurring. Now, over 2,000 participants in the US have been implanted with these permanent placed electrodes. And so taking a look at them, you wouldn't know that they have one of these there. Shown on the top right is an example brain activity, some data from this electrode, where you can see these brain waves and potentially what we're going to talk about, these theta oscillations, to try to bridge findings with rodents and measure this type of activity in the real world while they're moving around. So this is the first time we've been able to record deep in the brain while a participant is naturally moving, freely moving around. And I'll tell you today about some questions we've been asking to scientifically investigate how these regions are important for memory and whether we can translate some of these findings from rats and from the lab out into the real world. So here I'll show you a video that shows the setup where we have a participant with an example electrode that you see on the top right. It's implanted. We can put scalp EEG headset or cap, on his head, as well as a VR or AR headset and other wearables that can help us detect any kind of motion to synchronize with the deep brain activity. And so you see a setup here. It involves motion capture cameras. I'll be showing two videos here. The master. So in these videos, you see the setup of a participant with a brain electrode implanted who's navigating through a virtual environment that he can see on a VR headset. There are motion capture cameras that are tracking his position, his movement, his speed, and what he's seeing is updated in real time as he's walking through. And during all of this, we can record deep brain activity as well as stimulate to answer our scientific questions. All of this work that's been possible in these last few years through NIH funding required a multidisciplinary approach to make work. And what this means is that we need to work together with neuroscientists such as myself, as well as neurosurgeons who work with these patients and implant the electrodes, neurologists who are involved in the participants' treatment and help us to work with the devices that are implanted, engineers to make all of the wearable technologies, the VR headsets, and the deep brain implant synchronized and able to work together, as well as psychologists to help us with designing the tasks and computer scientists to help us program the tasks in VR and integrate it with all the wearable technologies. And then we also work with several industry partners, including the companies that develop the implanted devices, as well as virtual reality companies and companies that can consult with us on the various technologies. And of course, all of this is made possible through funding the Brain Initiative and several other funding sources as well. So this is what the setup looks like that we've developed. We call it a mobile deep brain recording and stimulation platform. There are several devices that together are used and carried in a backpack by the participant, to which we can wirelessly communicate in order to view the deep brain recordings, synchronize it with other data streams, and control it on demand. All of this work is done through patients and participants volunteering for these studies. This work is also posted online and as a preprint and including open source code that people can download. So this is what it looks like, the full version where you have full control, real-time recording, as well as programmatic control, wireless control of the implanted stimulator. This setup is also worn with an augmented reality headset in this case, a scalp EEG cap, as well as motion capture suit and biometric recordings that can record heart rate, respiration, skin conductance, and various other externally acquired data. We can also do stationary tasks, just like traditional studies have been done in the past, where a participant can sit down and look at a computer screen and do tasks in that way. We also have lightweight versions of the task, which involve wearing a device such as this on body, and the data cannot be viewed in real time in this case, but can be downloaded every 7 minutes or so. The wearable technologies that we use, like I said, include heart rate, respiration, skin conductance here, and you can also integrate it with wearable watches as well as eye tracking headsets, depending on what you want to do in the particular study. You can also take it outside and look at deep brain recordings during realistic behaviors out in the real world. So there's a lot of potential here with this platform to start asking questions that we haven't been able to do before in neuroscience because we've been limited by the immobility of our participants. Like I said, this paper is a preprint online and open source code is available so that other laboratories can replicate the setup and use it for questions of their interest. All right, so what can we do with this platform? What kind of questions can we ask? Well, for one, we have the ability now to wirelessly record and stimulate deep brain activity during freely moving behaviors in humans. So when we combine that technology with virtual reality, motion capture, and wearables, we can investigate, as I said, naturalistic human behaviors under experimental control. And last but not least, we can start to explore novel therapies that involve deep brain stimulation, neurostimulation, and begin to explore the neural mechanisms that are affected or that are involved in these type of treatments for patients with disorders. So my laboratory in particular is interested in episodic memory, as I've mentioned before, and so we sought out to use this platform to determine whether, for one, these phenomenon that were discovered in rodent studies are in fact present in humans. And this refers to theta oscillations, again, these 8 hertz oscillations that are important, that are thought to be important for memory and occur in rodents. But we do not know prior to this study whether they are present in humans during naturalistic behaviors, let alone, for instance, during movement. So are they present during naturalistic behaviors? Are they modulated by movement? Or are they modulated by memory demands? These are all questions we've not been able to answer until the development of this platform. And lastly, we decided to explore whether they are modulated by one's position in an environment, as you saw in the case of grid cells and place cells in the rodent, and whether in this case they are modulated in a grid-like pattern similar to the grid cells where they have a hexagonal geometric pattern to them across an environment. So these are some of the basic science questions that we sought out to answer with the platform. And I'll show you some of those results. So the first finding was that theta oscillations do in fact exist during movement in the hippocampus and entorhinal cortex, similar to the rodent findings. However, they're not continuous like they are in those studies. They're present in bouts and they come and go. So here just shows an example motion capture on the top left of a participant walking through the room. We had them walk around in circles and in straight lines. You can see on the top right the movement trajectory of an example participant through the environment. In the top middle, you just see the speed distribution. So we had them walk fast, we had them walk slow. So we had a variety of speed distributions across 4 sighted participants, and we also had a congenitally blind participant who navigated with a Hoover cane, and so the speed distribution of that participant is a little bit lower, but also varying quite a bit. And what we found was that movement that did in fact modulate the presence of these theta oscillations. You can see in the bottom left, 8 hertz oscillation signal that is significantly higher during movement compared to immobility. And the bottom right, you can see across time in an example data trace that these 8 hertz signals, they come and go. And so you see little hot spots of yellow as they are moving fast during periods of fast movement, shown in the gray trace that shows a higher speed in the beginning. And then towards the right, when they stop moving, you see the movement speed goes down, and those 8 Hertz theta oscillations are less prevalent. So this is, of course, one example participant. We have characterized this across the entire group of participants, and we found that statistically, these 8 Hertz theta oscillations are present, but they're more prevalent during the fast movement speeds compared to the slow. With the rodent studies, and in addition to the 16 Hertz harmonic that you see with the black line showing the areas of statistical significance, it was the case for both sighted participants and the blind participants that the faster walking speeds corresponded to a higher prevalence of these theta oscillations. But overall, the presence of these oscillations is quite low. If you look at the y-axis, it's about 10% of the time in the sighted, 30% of the time in the blind, which is much lower than we see in rodents. And so now we're exploring, why is this the case that it's so much lower, and what drives the increase in the prevalence of these theta oscillations? So we decided to do an experiment in virtual reality where we can control what the participant is seeing and viewing, and had them do a memory experiment where they had to, in this video that you see here, walk around and visit various locations that are outlined as a yellow cylindrical halo. But they have to learn the location of those halos and the order which they are presented, and later press a joystick whenever they reach those positions during a memory recall period. And so here in this video, you can see an example participant navigating the environment and learning these various locations and then having to recall them later. So this setup is all during simultaneous deep brain recording, which, again, as I mentioned before, has not been possible up until now. So the data we have here is from an example participant on the right. You see the top-down view of the locations that they had to learn in yellow, and they're recalled locations in the brown diamonds. And you could see after several blocks of learning, they do get better and they improve, such that on the right side in the last block, the positions of the brown diamonds correspond quite nicely to the yellow circles. And at the bottom, you can see overall averages across the group where they're learning the locations over time, over 5 blocks, and their memory is in fact improving on the position and the order of these locations. So they are learning these positions, and we wanted to next ask whether this type of memory experiment could modulate these theta oscillations, and whether there's any kind of spatial modulation in terms of the participant's position. Do we see any kind of grid-like activity in this data? And in fact, we do see a grid-like pattern in the data, which I'll show you in a second. But just to quickly explain how this analysis is done, based on the idea that these grid cells have a sort of alignment across them in this pattern you see on the top left, where the peaks of the grid cell firing fields have, if you're walking along their peaks, you should expect a higher level of activity shown in sort of peach color here, versus the gray areas where you are walking during positions that are not aligned to the grid cell activity. You shouldn't see as high of an activity. So this was the hypothesized effect that you should see if, in fact, theta oscillations are modulated by a grid-like pattern. And in fact, we do see that if we take the data and we split it for positions during which the participant is walking along the aligned area of the grid pattern versus unaligned in blue, you see a higher amount of theta activity during the aligned periods for every single participant that we tested in the study, including the blind participant who did a non-virtual reality version of the study and instead did an auditory task. So we found this finding all participants in a handful of electrodes that you see here shown on top of the dotted black line. So 5 electrodes showed this grid-like modulation of theta, and these happen to be in areas that we would expect, like the entorhinal cortex and subiculum. It did not, this hexadirectional modulation did not occur for other bands of oscillatory activity, such as beta or gamma, so it was specific to oscillation. And this effect was stronger after memory had been formed. So after they had learned the positions of the environment, if you take their peak slope of their learning rate and look at the hexadirectional modulation after that point compared to before, 3 of those participants showed a stronger effect after versus before. This one participant who didn't show the effect did not perform that well on this task, and so their learning rate was not quite as good, and so that may be confounding the results there. But it's evidence or suggestive evidence that memory could modulate this grid-like modulation of theta activity. And so we then wanted to look at other effects corresponding to theta activity across the environment. And we found another interesting finding whereby the position of the participant in the room modulated the theta activity, in that the closer they were to a boundary, the stronger the theta activity was. So proximity to the environmental boundary of the room, which in this case is the wall, modulated theta activity such that you had higher theta activity when they were closer to the walls compared to when they were farther. On the bottom left shows you an example from one participant, and on the right is the average across all of the participants in this study, where we see stronger theta power for boundary locations compared to inner environmental locations. And we also see this for the gamma frequency band, not just for the low frequency bands here. So here is another case where position in the environment can modulate theta power. And so the last question to ask is, does memory also modulate this finding? And I'll show you that in a second, but just to make sure, a couple other questions we asked, whether this effect was modulated by movement, and it turns out it was not. And so this effect occurred also during periods of standing only, shown in the middle, where you have stronger theta power for boundary locations compared to inner locations, even if they're just standing still and they're not moving. And on the right side, you see the effect also for areas where they are near, where they're not near a target location to be learned. So this is not dependent on movement, as shown in the middle plot, and it's not dependent on their position at a learned target location, which are 2 critical analyses to do to try to answer what really is modulating this theta activity with regards to boundaries. So the last question I mentioned was, is this effect, which occurs in every single participant, that's shown here on the top, P1 through 5, we have stronger theta activity for boundary locations compared to inner, is this effect modulated by memory? And it turns out it is. So when participants are looking for target locations, shown in green, you see the boundary effect. You do not see it for when they are walking to the walls in the case of the orange. So we have them in this task look for target locations in green in the center, you see in the center of the bottom left picture. But we also had them just walk to various wall locations. We had colored paper stimuli on the walls where we just had them walk to those locations. So there was no real memory demand in that case. And we found that this boundary modulated theta activity only occurred during periods when they were searching for the hidden targets, and not when they were just walking to the wall locations. In this case, we call this no target search. And you can see that broken up in the right panel on the bottom, where you see increased theta for boundary locations, for target search periods, and not for the no target search periods. So altogether, all of these findings suggest that memory can modulate these theta oscillations, and that these theta oscillations can be modulated by naturalistic walking behaviors, movement, and position within an environment close to boundaries, and also modulated by a grid-like pattern. So summarizing these results, high-frequency theta oscillations exist in humans during naturalistic behaviors. There's a higher prevalence of these oscillations during fast walking movements, perhaps related to a higher cognitive demand, which I didn't talk too much about here, but can discuss more if people are interested. And this theta activity is modulated in a grid-like pattern. It's stronger after a memory is formed, and it is also modulated by proximity to a boundary. And again, this effect is also stronger during periods of high memory demand and is independent of movement. So altogether, we've been able to start to translate findings from the rodent and move our laboratory-based studies into the real world. The next steps are, of course, to see whether stimulation can improve memory in the real world. And we can do that because this platform allows for stimulation on demand. And we can start to ask whether we can improve memory in the real world and hopefully contribute to future therapies for memory disorders. So I'd like to thank the team that did all of this work in my lab, postdocs and graduate students, programmers, and research assistants. And they all come from various backgrounds in terms of training and education. So it really truly is a multidisciplinary approach with neuroscience, engineering, psychology, and computer science. And also want to thank our research participants who volunteered, our alumni from the lab, Zara Agajan, who helped a lot with all of these studies, and the various other collaborators, as I mentioned before, and funding sources. So with that, I will take any questions if there are any, and thank you all for listening.